Many individuals think nothing of the simplicity of flushing away unwanted drugs, throwing rubbish into water sources, or dumping large amounts of toxic waste into Great Lakes. However, there is far more harm than benefit. What is the long-term impact of pharmaceuticals in our water supply? Some of the emerging pollutants detected are in fact responsible for the feminization of male fish on the Basque Coast and belong to the group of endocrine-disrupting chemicals. Chemically, they are very different from each other, but they have very similar effects. Due to their interaction with hormones, they destroy the hormone balance and can lead to the feminization or masculization of the organism. As they are pollutants, they have appeared recently. Little is known as yet about their effects on the environment and on the ecosystems. Despite the fact that they are new pollutants in terms of their effects, the sources of endocrine-disrupting chemicals can be found in everyday products: plasticizers, pesticides, contraceptive pills, fragrances, and detergents, among other things. Pollutants acting as estrogens are responsible for this phenomenon, which among other changes is causing ovocytes to appear in male fish. In fish, the effects of exposure of estrogenic pollution are diverse, and they particularly include effects on sexual development, reproduction, and behavior. Male fish exposed to estrogen produce vitellogenin, a protein that is normally only produced by females for deposition into developing eggs. Prolonged estrogen exposure during sexual development also feminizes the testes in males. In some effluent contaminated rivers, all male roach were found to possess feminized testes. In extreme cases, almost half of the testis can be made up of female tissues, developing eggs. These fish have reduced sperm quality and impaired reproductive performance in competitive breeding scenarios. Thus, at the individual level, the adverse health effects of exposure to estrogen are reasonably well understood. In contrast, effects of estrogen chemicals at the pollution level are less well understood. Findings that have raised the greatest concern, where long-term exposure have resulted in reproductive failure. To research this new phenomenon, bacterial transformation was conducted in the lab. The purpose of this technique is to introduce a foreign plasmid into a bacteria and to use that plasmid to amplify the plasmid in order to make large quantities of it. This is based on the natural function of a plasmid to transfer genetic information vital to the survival of bacteria. This way, scientists can get to the cause of the feminization of fish. In bacterial transformation, DNA transformation involves a transfer of naked DNA into the recipient cell. In the first step, double-stranded donor DNA binds to the DNA receptors on the surface of the complement cell. One strand of the donor DNA is degraded by nucleases while the other strand enters the cell. The single-stranded donor DNA pairs with a homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. If there are any differences between the nucleotide sequences of the donor and the recipient DNA's mismatch repair system comes into play. The repair system removes either the donor or the recipient strand and replaces it with a complementary sequence. Since either strand may be repaired, some cells contain the new donor DNA and others have the original DNA sequences. In the lab, cells are plated on selected media so that only the transformants will grow. Bacteria reproduce by binary fission. The bacterial chromosome is circular. The replication process starts at the site called the origin, but instead of replicating in one direction around the chromosome, it replicates in two directions. This is called bidirectional replication, as two replication forks move in opposite directions away from the origin. The forks will meet at the bottom of the chromosome, and replication is terminated as the two chromosomes separate. The bacterial chromosome is also capable of initiating multiple replication forks. Mutations are the cause of the feminization of fish. A point mutation is a change of the nucleotide sequence of a gene. This can result from an error during replication that is not corrected by the usual repair mechanisms. Under normal circumstances, adenine and thymine should bind to one another, as should cytosines and guanines. In a base substitution mutation, a non-complementary nucleotide is incorporated into the new strand. The resulting altered triplet may code for a different amino acid or even a stop codon. Here we see 4 codons and their corresponding amino acids. A change within the second triplet results in a change in the amino acid. None of the other amino acids are effective. Insertions and deletions result in frame shift mutations. The insertion of an extra nucleotide not only affects the triplet it is part of, but also the subsequent triplets, thus drastically altering the protein product. The result is similar if a nucleotide is deleted. The feminization of fish is a widespread phenomenon solely caused by human error and laziness. This can easily be avoided if we act responsibly. The effect of this could cause other species of animals to die out as a result of the overpopulation of female fish, disrupting the natural flow of the food web.